Hello, my name is Logan Readnour. I am a second-year grad student in the Ag and Bioengineering department, working for Dr. Kevin Solomon, and today I'm going to tell you about the technology we are developing, which are tunable synthetic transcriptional regulators that are responsive to environmental triggers. So why do we want to do this? Well, one of the aims of synthetic biology is to be able to develop microorganisms that can produce useful products such as chemicals, biofuels, or drugs by making changes at the genetic level. And this type of work can have a very large impact. For example, researchers at UC Berkeley were able to genetically engineer yeast to produce precursors to artemisinin, which is the most effective treatment for malaria. So their partnership with Amyris and Sanofi led to the large-scale development of semi-synthetic artemisinin, and they now produce 1/3 of the world supply of artemisinin, vastly improving the affordability and availability of the drug. So a current challenge in the field right now is that the buildup of toxic products in these bioproduction pathways often leads to lower yields due to adverse effects on cell health. So therefore, there is a need for programmable regulators that can be universally applied to multiple pathways to auto-regulate gene expression of these toxic products for improved cell health and production. And this is exactly what our technology aims to do. So we are developing novel transcriptional regulators by fusing elastid-like polypeptides, or ELPs, to sigma factors. And we are taking advantage of the reversible inverse phase transition property of ELPs, which means that they are able to aggregate or become soluble based on changes in temperature or pH. So the availability of sigma factor will then, to turn on gene expression, will then depend on the aggregated or soluble state of ELP. So there are several advantages to our technology. The first is that the ELPs are programmable, meaning that we can make specific changes in the ELP sequence to change the transition property. So we can therefore make an entire library of ELPs that transition under various conditions. And the second is that we can have targeted regulation of specific genes, and this process does not interfere with the native processes already within the cell. And lastly, we can expand the regulation of toxic products. So we do not have any third-party obligations, and a provisional patent has been filed by the Office of Technology Commercialization at Purdue. So if you have any questions or would like any more information, please contact OTC for more information.